Good evening, everyone. So, today we will start with tutorial session 9. I will share my screen. My screen is visible to everyone, right? Okay, thank you. Okay, so last week, both the weeks we looked at chromatographic techniques. How will you really purify your product of interest or sample of interest? And now you have to characterize your product. So we did look at gel filtration chromatography or SEC, wherein we were using the peaks, like where the elution, where our sample of interest or the protein of our interest is eluting at which particular volume, then we could tell what is the oligomeric status of the protein, how much might be the range of the uh, like molecular weight. But then just by looking at the chromatogram, we won't be able to tell that okay, this is the exact molecular weight. So by like using SDS page, using native page, and all those techniques, all those electrophoretic techniques will additionally help in characterizing the product of our interest. Okay, so one way to characterize the product is by looking at the molecular weight, where we briefly looked at gel filtration chromatography, wherein we get the native molecular weight of the protein, or we can also get like get the status of the aggregation or oligomeric status of the protein. Electrophoresis, we can get the native and subunit molecular weight of the sample of our interest, basically protein here. And mass spectrometry, we can get mass spectrometry is uh, like a kind of foolproof technique, wherein we will get the exact molecular weight of our molecule of interest, sample of interest, whether it can be protein, nucleic acids, or any metabolite. Okay, if something is attached to our protein, some kind of ligand is attached. If there are in mass spectrometry, you can also have results, like as in you can also characterize or distinguish between uh, proteins that are post translationally modified. Okay, that those modifications can also be determined using or detected using mass spectrometry. Okay, functional groups, we can get an idea using infrared and UV visible spectroscopy. These will be, I think, in the next week's lecture, which also gives an idea about the native and subunit molecular weight. And then you can definitely sequence your sample. So you get the peptide sequence, or if it's a plasmid, suppose you have isolated plasmid. Now, if you or you have cloned your uh, gene of interest into a certain vector, particular vector. Now you need to know, okay, what is whether the ligation has taken place. So we send our samples for sequencing, wherein you get the exact um, nucleotide sequence, and then you can always align the sequence and just look at your results and interpret whether the gene of your interest has incorporated into the vector or not. This is regarding nucleic acid one. Peptide sequencing can also be done in case of proteins. So you can always check that whether the sequence that you have obtained from the sequencing results match those of your protein of interest or not. Okay. Now, this is what using gel filtration chromatography, we can determine the molecular weight, wherein the largest sized molecule will elute like fastest, will elute the fastest as compared to the smaller uh, sized proteins or smaller sized molecules. And then uh, it is basically related to the shape of the molecule, and relationship between molecular weight and radius of gyration is this. We looked at this in the last week's lecture as well. Okay, then we can determine the oligomeric status of the protein, because Here you will get certain peaks. Okay, and but on SDS page, because SDS is a denaturing page, you add reducing agent like BME, beta mercaptol or DTT, or you are adding detergent, which will hamper the inter-subunit interactions taking place in a protein. In case the protein is oligomeric, like suppose it's dimer or a tetramer or a trimer, okay, so those interactions will be hampered in a reducing uh, electrophoretic gel like SDS page, and then you can determine the oligomeric status of the protein of your interest. Okay, so this is how we can look at the oligomeric status. Molecular weight obtained using gel filtration, because this particular elution volume uh, will give now these elution volumes are characteristic of the particular type of column that you are using for SEC. Now based on that, they have each particular kind of matrix or the particular column, based on its dimension, based on what kind of matrix is packed inside, all these properties determined, like they are like especially the pre-packed column also come in with a manual wherein you have all the range, like suppose certain molecule is eluting at this particular volume, then what might be the range of the molecular weights that will elute out at this particular volume for a particular type of column. Okay, so based on that, you are getting a molecular weight, and definitely while running on SDS page, you are having molecular markers like reference protein standards, protein which we call as reference or standard or ladder. Okay, protein ladder in case of proteins. So, and otherwise, in case of nucleic acids, we use DNA ladder or RNA ladder. Okay, we will eventually look at this in the subsequent slides. So based on that, you can make out that whether what is the oligomeric status of the molecule of your interest, okay, or sample of your interest. We can study protein folding, right, using urea, and like how much your protein has denatured. We can always infer from the peaks like chromatogram. The completely unfolded protein will elute the first, okay, will elute the fastest because of its larger size or overall hydrodynamic radius. It won't have access to the pores of the matrix in a gel filtration column, and then it will come out, elute out first, followed by the very well folded protein or the native structure of the protein that will have comparatively smaller hydrodynamic radius, right? And it will elute at the end as compared to the other partially unfolded or completely unfolded protein. Okay, and then we can study whether there is any interaction between a protein, if we want to study if there is any ligand that is binding to our protein, or suppose we need to study, suppose like there are calcium ions, I need to study some calcium binding proteins. Now I need to know that how many pockets for calcium binding are there. Okay, there are other biophysical techniques definitely. So so there are, there's ITC or calorimetry techniques, like as soon as the ligand is binding, there will be energy changes, whether it can be exothermic, like energy can be released, or it can be endothermic, energy can be taken in to perform a particular reaction, for a reaction to happen. Okay, so based on the interactions of the like either it can be protein or ligand or protein interactions. Okay, suppose there are like many processes, it's like suppose cell division machinery or replication or transcription or translation. So all these pathways have numerous proteins uh, playing roles in at different steps, right? So what can happen, like how did people like know that okay, these are the key players in this particular pathway? Suppose they want to study the interaction, because multiple factors can be coming together and then performing a particular reaction, right? So for studying the two reactions, or for studying, or for like studying all the factors that are coming into form a complex, and then playing their individual roles after forming the complex in a particular pathway or in a particular type of metabolic pathway, then what you do is you can always look at the complex formation using SEC or gel filtration chromatography. Okay. And now, so this week's content had, I mean, the week 9 content had electrophoretic techniques. So there are different types of electrophoretic techniques. We will eventually go through it one by one. So electrophoresis, it depicts or is a term that refers to the migration and separation of charged particles or ions in an electric field. Okay. An electrophoretic framework has two terminals, definitely one positively charged and negatively charged electrodes, which are separated or associated by a directing medium called an electrolyte. Okay, because these will enhance the movement of ions, right? These movement of ions will eventually help in the movement of your sample of interest along a particular direction. Now, the separation impact on the ionic particles results from contrasts in their speed, which is the result of the molecule's portability or, in general, molecule's mass, definitely the size of the molecule, and the field strength, how strong voltage or how strong is the electric field applied. Based on that, the movement of the molecules is like movement of the molecules is affected. Now, suppose this is the direction of the electric field. So, this is the electrostatic force experienced by, uh, ex suppose this is your molecule of interest. And this is the overall charge on it. And this is the electrostatic force. Definitely, if the electric field is towards this side, this is the electrostatic force. And definitely, if something is moving through a medium, okay, whether it's a solid medium or a liquid medium, there will be definitely a retarding force or a frictional force towards the opposite direction of its movement. So there will be friction force and electrophoretic retardation force. Okay. Now the portability of an ion not entirely set in stone by molecular size, shape, and charge, and the temperature during the separation, and is consistent under characterized electrophoretic conditions. Okay. So this is from the lecture video only. So this is the field direction, electric field direction, the same thing. This is the force being experienced by your sample of interest, and this is the frictional or the opposite force or the frictional force acting on this particular molecule. So overall, the mobility is affected by when we get this equation by solving all these, equating it at a particular time point. Then what we get, the mobility or V is directly proportional to the charge on the particular sample of interest or molecule of interest at a particular time point, and inversely proportional to the viscosity of the medium. Definitely, the higher the viscosity, the slower will be the speed. Okay. What is the size and shape of the molecule? The greater the size, the slower will be the speed again. And in the case of relative mobility, it is directly related to the charge by radius of the molecule. Okay. So for a globular protein, the radius of the molecule is related to the molecular mass of the macromolecule. So relative mobility is basically charge upon mass. Okay. Now, this is overall like what is going on or taking place in a like inside an electrophoretic tank. Okay. And what are the different forces being experienced, or what are the forces that affect your sample of interest when it is moving through uh, and like moving through an electrophoretic setup. Okay. Now, there's moving boundary electrophoresis and zonal electrophoresis. Okay. So, moving boundary electrophoresis, as the name suggests, that it doesn't have any solid or support matrix. Okay. The, the electrolyte or the thing that is there is moving. Okay. So, what happens here is this is carried out in a U-shaped tube, this one, with platinum electrode attached to the end of both the arms. Okay. At the respective ends, a refractometer is attached. Refractometer actually monitors the differences in the refractive indices. Okay. Now, sample is dissolved in the buffer and is loaded in the middle of the tube here. Okay. Then the apparatus is connected to the power supply. So what happens is, because power supply is there, there's an electrolyte in the whole setup, like there is electrolyte present. The movement of ions will help in the movement of uh, ions across the electrodes, right? And that will ultimately help in moving your sample as well. Sample will also be having some intrinsic charge on it, okay? Or based on what type of electrophoresis there can be, like in SDS page, due to the presence of SDS, which is an anionic detergent, your protein is coated with negatively charged SDS particles. So overall, there is like negative charge deposition on the protein of your interest. Okay. So based on what type of electrophoresis you are carrying out, if it is native based, then it will be solely depending on the intrinsic charge of the molecule of your interest, right? But like coming back, in moving boundary electrophoresis, the charged molecule will move to their respective to its respective counter charged or oppositely charged electrode and passes through the refractometer, and refractometer will measure the change in the refractive index of the buffer due to presence of the molecule, right? Now, as your sample of interest or your molecule of interest passes, the sample is taken out from the apparatus along with the buffer. Okay. Now, this is used to study the behavior of the molecule in an electric field. But the thing is, the resolution of this particular technique is very low, because things are like, from here only things are passing towards this electrode as well as this electrode. So there is mixing of the sample. Okay, suppose there are multiple samples you need to study or you need to check. There is no separation between the samples. And the resolution is very low because there will be intermixing of the samples. So, like one particular sample might have some uh, suppose we are talking about a protein, maybe, so okay, or a nucleic acid mixture, suppose. So then one kind of thing will be having similar charge, one kind of subunit will be might have similar charge, so it will move towards that side. So there will be like the resolution is too low and there will be intermixing of the sample, okay, or overlapping of the sample components. And then there's zone electrophoresis. What happens here is, it involves the migration of the charged particle on a stabilizing media or a solid matrix. Okay. So here you see, like in agarose gel electrophoresis, there was a demo in one of the lecture videos also. This is your gel, this one. Okay, this is the tank, this whole yellowish, and the tank is filled with buffer, and this whitish bluish thing is your agarose gel. Okay, the tank has two electrodes connected at both the ends. And suppose in case of nucleic acids, if it is a nucleic acids, then DNA, or suppose nucleic acids, due to the presence of phosphate groups, are negatively charged. So what happens is, see these marks here, these are the wells wherein you load your samples, you put in your samples here, and these are the slots for samples, we call it as wells. Okay, and the tank is filled with appropriate buffer, which is having electrolytes which will move from one electrode to the other. And then when you load your sample here, what will happen is, especially in case of nucleic acids, this is negatively charged. Your nucleic acid is negatively charged, and this is negatively charged electrode. So there will be repulsion, and and also this negatively charged nucleic acid will move towards a positive charge, like positively charged electrode, thereby, but then it will again, the movement based on how big the sample or how big the molecule is, based on that it will be separated, because there will be a sieving effect on the sample of your interest, wherein it will separate the sample components like based on the sizes or molecular weights. Okay, so the components separated are distributed into discrete zone in the supporting media or the supporting matrix. Supporting media is saturated with buffer solution. Small volume of sample is applied as narrow band. And on application of sample at the ends of the strip, components migrate at the rate determined by its electrophoretic mobility. Separated colored components can be separated by electropherogram. Okay, so there are different kinds of zone electrophoresis, wherein the solid support or stabilizing media that is being talked about here, that can vary. Okay. because it can be paper electrophoresis, or it can be agarose gel electrophoresis, it can be polyacrylamide electrophoresis. Okay, so there the depending on what is your sample of interest, what is your requirement for the experiment at hand. Based on that, you can use whatever supporting media or stabilizing or solid matrix you need to use for electrophoresis, you can use that. Okay. Now we looked at SDS page electrophoresis, but we will again look at it one like in detail. So what happens is for SDS page in SDS page electrophoresis, you have two kinds of gels. Okay, definitely there are two electrodes, one negative charged and positively charged electrode, and in SDS page, because the protein or sample of your interest is coated with SDS anionic detergent, so it will have a negative charge, right? So it will move towards the like will be repelled from this negatively charged electrode, and it will move towards the positively charged electrode, right? And it has two kinds of gels. One is the resolving gel, wherein the actual separation of your molecule of interest happens. It is at a particular pH of 8.8, and there's the stacking gel above the resolving gel, which is of a pH 6.8. Now we will eventually again study what is the significance of these pH values. Okay. Now in general, if we look at yeah, this is the direction of the electrophoresis, like from top to bottom, and these are the wells, these ones, okay, wherein you load your samples. And so what if we what is happening here, this is a matrix which is having a di different kinds of pores depending how much of your like polymerizing agent has been put into the solution. Based on that, the pore size will vary, and this is your mixture of molecules, macromolecules have been put here, your sample that is there, and then it will have a sieving effect, and it will go through it based on how bigger the thing is. See, the pinkish pink molecules here are really small, so it will move the fastest, and the yellow molecules here are the largest ones, so it will take time to go move through the pores of the matrix. Okay, so how SDS page works? SDS page separates proteins according to their molecular weight based on their differential rates of migration through a sieving gel or a sieving matrix under the influence of an applied electric field. Okay, and movement of any charged species through the electric field is determined by its net charge, the molecular radius, how what is the size or the hydrodynamic radius, and how strong is the applied electric field. Okay, how much voltage have you applied, or what is the potential difference. Depending on that, the speed of or the movement of the sample or the macromolecule of interest uh, differs. Okay, and in case of natively folded proteins, what happens is neither their net charge nor their molecular radius is molecular weight dependent, right? Because uh, most of the when protein is folded in its native form, it is having a particular structure, right? So many of the the overall radii or hydrodynamic radius, that is the resultant radius or resultant size of the natively folded protein, will definitely differ, as well as the charge will also be whatever the surface charge is, that will also differ based on what kind of amino acids are there uh, on the surface. Okay, because most of the hydrophobic amino acids will be uh, again depending on what kind of environment is, what kind of solution is there surround like your protein of interest is in, like uh, what is the solution the proteins are inside, depending on that what kind of residues are outside or inside in a hydrophobic core, or whether the surrounding medium is hydrophobic, then your hydrophobic residues will be outside. Okay, so based on what kind of solution is, what is the surrounding medium, the net charge and the molecular radius will be varying. Okay, for a natively folded proteins, their net charge and what actually gives them the charge, different kinds of charge on a protein, it is amino acid composition, because amino acids are the like based like owing to the R groups or the functional groups, different kinds of functional groups. Protein amino acids can be positively charged, negatively charged, uh, like can be polar, non-polar, okay, or can have hydrophobic residues, or it can like either make hydrogen bonds, it won't make hydrogen bonds, depending on it will make hydrogen bonds because it definitely has NH2 and carboxy terminal, but depending on what how much of the those hydrogen bond forming atoms are exposed, or how much it is buried inside, depending on that again the hydrogen bonding interactions or other inter as well as intramolecular interactions will vary. Okay, so in the native state, different proteins with same molecular weight can migrate at different speeds in an electric field depending on their charge and 3D shape. Okay, now to separate proteins in an electric field based on their molecular weight only, we have to destroy their tertiary structure by reducing the protein to a linear molecule, and we have to mask the intrinsic net charge of the protein. Okay, because this will hamper with the mobility of the protein. Okay, there won't be any uniformity, so we can't really be conclusive about, suppose if we want to look at the molecular weight, right, we can't have a conclusive result on that. Okay, now let's look at for SDS page what are the different kinds of reagents being used. Okay, the different buffers and reagents being used are timid. This is NNN tetramethyl ethylene diamine, which catalyzes acrylamide polymerization. Okay, so the polyacrylamide that is there, it is a composition of acrylamide and bisacrylamide. Okay, acrylamide uh, is the linear branching, but the sorry, linear polymer like linear polymers or polymerization is takes place using acrylamide, and the branching between two linear chains takes place due to bisacrylamide. Okay, now timid, this particular catalyst actually speeds up the polymerization process. Okay, and there's APS or ammonium persulfate, which initiates or which provides radicals for acrylamide polymerization. Okay, then there's tris HCl, which is the component of running and gel casting buffer, and the buff there can be it can be of different pH. Okay, so overall in the um, SDS page electrophoresis, we have three buffers. One is the running buffer, okay, which is usually tris glycine SDS buffer or TGS buffer, okay, and you have the stacking gel, like stacking buffer, right, and the resolving buffer, stacking buffer with pH 6.8, resolving buffer with pH 8.8, okay. You have glycine, which is the component of running buffer, and bromophenol blue is there, which is the tracking dye to monitor. This is used for your sample preparation, which is the tracking dye to monitor the progress of gel electrophoresis. Then there's Kumasi brilliant blue, which actually binds to the protein of interest, so that we can detect that okay, our protein of interest is at this particular point on the gel. Okay, but this Kumasi brilliant blue will come into the picture after the gel run has completed. Okay, because you have to visualize the gel, right, wherein where is your protein of interest, okay, located at which point, and then you compare it using the reference or the standard or protein ladders that are available, and then determine that okay, this is the molecular weight of the but this particular band. Okay, then there's SDS, we have talked about it multiple times, which is an anionic detergent. It is used to denature and provide negative charge to the protein, because we in the previous slide we talked about we need to remove the intrinsic, not remove, we can't really remove it, we can mask the intrinsic charge of the protein. So SDS does this work wherein it covers the or coats the protein uniformly with a negative, imparts a negative charge to the overall protein. And there's acrylamide and bisacrylamide. Acrylamide is the monomeric unit used to prepare the gel, and bisacrylamide is basically the cross linker for polymerization of acrylamide monomer to form this particular solid support or the gel. Okay, see here, this is acrylamide, this is bisacrylamide, and timide is the catalyst, and ammonium persulfate supplies the free radicals. Okay, so this is how the polyacrylamide matrix looks like. Now, ammonium persulfate in the presence of timide, the catalyst, forms oxygen-free radicals and induces the polymerization of acrylamide monomer, this one, to form a linear polymer. And these linear monomers are interconnected by cross-linking using bisacrylamide, okay, to form a 3D mesh with pores. And the size of the pore is controlled by the concentration of acrylamide and the amount of bisacrylamide in the gel. So usually for like, for the daily in protein laboratories, for regular running of gel, or like the usual we use for the usual SDS page electrophoresis, we use around 30% acrylamide, uh, but wherein it has, I think, in 19 is to 1 ratio of acrylamide and bisacrylamide. Okay, like lot of acrylamide is there, and accordingly a small amount of bisacrylamide is there for the branching or cross-linking of the acrylamide polymers. Okay. Okay, this is the scenario how it actually looks at the molecular level. Now, SDS is a detergent present in the sample buffer. What happens is it is also present in the running buffer. It is TGS, glycine, and tris, glycine, and SDS. And in sample buffer, what happens? You also, like, the sample will be running through the gel definitely, but then you have to prepare the sample also. So you add certain kind of dye, right? We looked at bromophenol blue and all those dyes, which helps in tracking how much your sample has run, how far the sample has run on the gel, okay, or how long do you have to run the gel so that you can visualize your sample of interest properly. So SDS is one wherein you prepare, like you add it in the loading dye, okay, and then what you immediately do, like sometimes SDS cannot always denature the whole protein, okay, so what is recommended that after adding SDS you have to immediately boil the sample, and the loading dye also has a reducing agent like DTT or beta mercaptoethanol to break down disulfide bonds. Okay, so all these detergents, this boiling and the presence of reducing agents disrupts the tertiary structure of proteins, and the folded proteins are linearized. Okay, folded proteins are linearized. So SDS coats the protein with a uniform negative charge which masks the intrinsic charges on the functional groups or R groups, and it binds quite uniformly to linear proteins. Around 1.4 gram SDS binds to 1 gram of protein, which is almost the same level, right? Almost similar, meaning that the charge of the protein is now approximately proportional to its molecular weight. Okay. Now, the dominant factor in determining SDS coated protein's mobility will now be the molecular radius. Okay. So SDS coated proteins are shown to be have been shown that it has been shown that those are linear molecules which are around 18 angstroms wide and with length proportional to their molecular weight. So the molecular radius and hence the mobility in the gel is determined predominantly by the molecular weight of the protein. Okay. So now this is what I was talking about. So this is the running buffer, tris glycine SDS TGS pH 8.3. This is the stacking buffer or the stacking gel, which is tris HCl pH 6.8. This is this dark gray one is the resolving gel. So stacking gel has low concentration of acrylamide where we actually don't separate the proteins there, and then there's running gel here which has higher concentration of acrylamide wherein like the size of the pores will matter here because that will provide the sieving effect. Okay, here we don't want any separation, we don't want sieving of the thing, so pores should be good enough so that almost all the proteins can easily settle down or move through here. Okay, and then come to this starting point, like we will eventually look at it. And this running gel or resolving gel has higher acrylamide concentration capable of retarding the protein movement. Okay. So the stacking gel here which is having a pH 6.8, what happens here is, okay, before that, uh, an amino acid can exist in a, it has NH2 group, it has carboxyl group, okay, which can exist as COO minus, and NH2 group can exist as NH after accepting uh, or after donating its lone pair on the nitrogen atom, it can exist as NH3 plus, right? So positively charged, negatively charged. And again, additional charge can be um, given, okay, additional charge can be there due to the type of functional group present in an amino acid, right? So in case of glycine, it has H, okay, H, H, NH2 and COH, right? So this is the glycine. So depending on what is the pH of the surrounding medium, the pH can be, so as like as if the pH is low, then H plus concentration is more in the surrounding medium, right? So if H plus concentration is more, COO minus will have will be existing as COOH. But since the H plus concentration is more in the surrounding medium, the lone pair on the nitrogen atom can easily donate its electron pair to one hydrogen ion, right? And then form NH3 plus. So at lower pH, glycine exists as positively charged amino acid, right? But then slowly when the pH increases, okay, what happens is, uh, as the pH is increasing, H plus ion concentration is decreasing, and because CO minus has a higher tendency to being acidic, it has higher tendency to donate its electron. Sorry, donate its H plus. Okay, so what will happen is this will give away its H plus, thereby forming CO minus, and NH3 plus is there. So the net charge or the overall charge on this amino acid at this particular point is 0, right? So this is the Zwitter ionic state of glycine at this particular point. So Zwitter ionic, and this the pH at which the amino acid exists as a Zwitter ion or as a neutrally charged molecule, that is called the Pi or the isoelectric point. Pi is basically the pH at which this particular amino acid is existing as a neutrally, where the net charge on the amino acid is 0. It's not that there are no individual charges present. Charges are there, but the net charge on the molecule is 0. What will happen is, in its Zwitter ionic state, that particular amino acid won't be moving anywhere because like, it is having under the electric field, so it won't be having any preference towards any electrode. Okay, but eventually, as you increase the pH further, H plus ion concentration is too low now. OH minus is higher, right? So what will happen is H plus is go away from the H plus will go away from the NH3 plus group, like NH3 plus moiety, thereby forming NH2, and CO minus will definitely has already given its H plus away. So the overall net charge on the amino acid at this particular pH, at a higher pH, is negative. Okay. So depending on that, so in case of glycine, which is the simplest amino acid as compared to the other 19 amino acids present, uh, these are the three charged states of glycine. Okay. Now, what happens is at pH 6.8, glycine switches predominantly to the Zwitter ionic or neutrally charged state. This is the one, the middle one. Okay. It has no net charge on it. So the loss of charge causes the glycine molecules to move very slowly in the electric field, right? Because it doesn't have any preference towards an electrode. So it will either move due to the dragging of, or suppose some kind of other molecules are moving ahead of it or above it, then that dragging force will help in moving the glycine down. Okay. Other than that, there won't be like it, this particular neutral charged molecule won't be having any preference towards the positive electrode, right? Now what happens is, this is tris Cl. Now the chloride ions from tris HCl will move much more quickly in the electric field. First of all, it's negatively charged, and also it's too small, right? Too light, so it can easily move, and it will form an ion front. So here just we are focusing on this particular part, the light gray part, the upper part. Okay. It will move much more quickly in the electric field, form an ion front, migrating ahead of glycine definitely, and separation of chloride from tris counter ion, which is now moving towards the anode, creates a narrow zone, okay, with steep voltage gradient that pulls the glycine ions along behind it, resulting in two narrow separated fronts of migrating ions. The very first one will be highly mobile chloride front, followed by the neutrally charged glycine amino acid front. Okay. Now, all the proteins in the gel sample will have an electrophoretic mobility that is intermediate between the extreme mobility of glycine, which is really slow, and chloride ions, which is really fast. Okay. We are talking about the mobility here along this particular setup. Okay. Now these two fronts sweep through the sample well. Okay, this sample well, everything is having this particular pH. Okay, 6.8. Two fronts sweep through the sample well and the proteins are concentrated into the narrow zone between chloride and glycine ion front. Okay. Now, this procession carries on until it hits the running gel. This part. Okay. And the pH switches to or changes to 8.8. Now, what we saw here, see, as the pH is increasing, the glycine amino acid becomes negatively charged. Okay. So the pH switches to 8.8, glycine molecules are mostly negatively charged and it migrates much faster than the proteins. Okay. So now what will happen is chloride ions are anyways moving ahead. Okay. Being due to its small size as well as negative charge on it, chloride ions are moving, but the glycine molecules which were like moving really slowly, now they are having a net negative charge on it, so they will move past proteins, right? Now glycine front accelerates past the proteins and proteins are dumped in a very narrow band at the interface of the stacking and running gels at this particular point. Okay. Now, so they will have a starting point wherein all the proteins are stacked at the same level, and then they will experience the sieving effect due to the higher polyacrylamide concentration in the resolving gel or the running gel. Okay. So the running gel has increased acrylamide concentration. It will slow the protein movement and then the sample or the proteins will be separated according to their size. So why at all the stacking gel is needed? Okay. So what is that? The gel wells are nearly 1 centimeter deep. These ones. Okay. These ones wherein you will be loading your samples eventually. So we need to substantially fill them to get enough protein for visualization. Okay. Now, absence of a stacking gel, what will happen is sample would sit on top of the running gel as a band of up to 1 centimeter deep. Okay. Now, rather than being lined up together and hitting the running gel together, proteins would all enter the running gel at different times, resulting in very smeared bands. Okay. So, stacking gel ensures that all the proteins arrive at the running gel at the same time. So, proteins of the same molecular weight will migrate as tight bands. Okay. So, there are different kinds of uh, gels, like 7% having different concentrations of acrylamide. The higher the concentration of acrylamide bisacrylamide, the smaller will be the pores. Okay, and like it will have like it will be able to separate out the larger proteins or larger sized proteins would be moving really slowly. So if you know that your proteins size or whatever sample is there, it will have your molecules ranging having like really higher molecular weight, then the idea is to have less concentration of acrylamide and bisacrylamide in your SDS page gel. Okay, so that it moves, it has enough uh, like like enough space through the pores to move and separate out. Okay. And but in case the molecules or protein of an interest is really small, then there should be higher amount of acrylamide percentage in the gel so that the pores are smaller and you can separate out the bands properly. Okay. It doesn't like really move through the gel and just go out of the gel altogether. Okay. Now, we will talk about the loading dye. We talked about it a bit, like we were adding SDS, we were adding reducing agent in the loading dye. Okay. So loading dye is a sample buffer used to prepare and load protein samples onto SDS-based gels. It contains a number of components. SDS, we talked about it, it denatures proteins and adds a negative charge, allowing them to move in an electric field. There's bromophenol blue, which is the tracking dye, which allows you to visualize the proteins as they run on the gel. So visualize the proteins, like not really the proteins we are visualizing. What is happening is bromophenol blue has like it runs at like speeds like really fast. It runs really fast. So what will happen is there will be a bromophenol blue has bluish colored dye front. Okay, so it will move the fastest. Now based on that you will understand, okay, this is how much my gel has run, so maybe it can run a bit more. Okay, depending on that, this is a tracking dye, right? Then there's glycerol. What happens is it increases the density of the sample, making it easier to load so that it settles your samples, gets inside the wells properly. Tris, we saw, it helps maintain the pH. Then there's reducing agent like dithiotretol or BME, beta mercaptoethanol, which breaks the disulfide bonds and keep the proteins unfolded. Okay. Now, after your gel has run and everything, so bromophenol blue is the tracking dye. Okay, but now you have to really visualize, okay, where is your protein of interest located on the gel? So there's Kumasi brilliant blue, a particular kind of stain that binds to proteins, okay, or binds to amino acids. So what happens is this is Kumasi brilliant blue is used for routine visualization of proteins which have been separated or resolved on polyacrylamide gels. So this is an organic dye that makes complexes with basic amino acids like lysine, histidine, tyrosine, and arginine, thereby giving an overall negative charge to the proteins, allowing their separation on the polyacrylamide gel. This is also, yes, this can also be added during separation as well as it can just be added for visualization. Okay. Different kinds of Kumasi dyes are available. It can be brilliant blue R250 or G250, where the R stands for more red and G stands for more green in the dye. So what is the principle of staining? When we talk about the staining thing, what is the principle? So this particular dye has three forms: anionic form, which is bluish in color, neutral form, which is green in color, and cationic form, which is red in color. Okay. Now when the environment is acidic, the red dye is converted into its blue form, this anionic form, after binding to the protein of interest. Now, if no protein binds to the dye, then the solution will remain brown. Okay, and this Kumasi brilliant blue stain forms a strong non-covalent complex with the carboxyl group of the protein by van der Waals force and the amino group through electrostatic interactions. Okay, now when this complex is formed, the red form of the Kumasi dye first donates its free electron to ionizable groups on the protein, thereby disrupting the protein's native state and exposing its hydrophobic pockets. And these pockets non-covalently bind to the non-polar components of the dye via van der Waals force. The ionic interaction further strengthens the bond. Okay, and this, due to this particular bond formation, the complex, due to this complex that has been formed, it helps us to detect the protein bands on the gel. Okay, see, when you stain with the Kumasi brilliant blue dye, this is like totally blue or purplish kind of. Okay, it's like you can't really see anything, but eventually when you destain it, you can use methanol, glacial acetic acid, or like it will destain the things faster. So because your proteins are, see, the whitish part doesn't have any protein, so the dye didn't really bind, but these are the protein bands, and because of the complex formation that we talked about in the previous slide, this is much tighter, so the dye doesn't come off that easily, but the other areas the dye comes off really fast. Okay, now due to, and then you can visualize where the protein bands are present on the gel. Now also during this case, sometimes this stain that we use in protein labs, what happens is this Kumasi brilliant blue dye this particular gel staining solution that we prepare can be used multiple times. Okay. So what many labs do, that multiple times the solutions can be used. So because over the time it can also happen that the efficiency of this dye binding to the proteins decreases. So people can also increase the efficiency by heating. Like as soon as you have like your gel has run, you have transferred your gel from the plates like SDS gel running plates, like from the apparatus. You transferred it, you put on, like dipped it in the staining solution, and then you heat it a bit. Okay, so that you really fasten the process of this dye binding to the proteins. Okay, and so it will stain your gel or stain the protein bands much more efficiently, and then you can destain it again. Okay, now, okay, this is how you have visualized. You can see your protein bands now. Now, how will you determine the molecular weight of your protein of interest or sample of interest? Okay, so this is now in case of protein, there is a standard marker, okay, having different kinds of uh, different molecular weights here. Okay, this is the marker protein having different types of protein fragments or peptide fragments here of different sizes. So what you do is you calculate this particular value, which is the ratio of mean distance traveled by the protein of your interest. Okay, this is your sample divided by the distance traveled by the dye. So dye means the bromophenol blue or the tracking dye we have been talking about. The tracking dye will form a dye front here. So with reference to that particular dye, how far your sample has moved, we can calculate the ratio. Now, first of all, we have to calculate the individual ratios of each peptide segment here in the marker lane. Okay, suppose there are 5 fragments here. So each 5 fragment will have a certain movement along the gel. Okay, these are like standard, these are of standard sizes. And with reference to the dye front, each fragments in the marker lane, each marker fragments RF value or RF ratio will be calculated and plotted on the graph, right? And then you will, like based on the curve, now you will be, like based on the samples distance here, okay, it will have a certain value, the RF value. Based on that, you can like extrapolate it using the standard curve and determine your protein of like determine the molecular weight of your protein of interest. Okay, now this is how you will be determining the molecular weight of your protein of interest. Okay, now the we talked about SDS page. Okay, but SDS page is a denaturing kind of electrophoretic analysis, right? So, but you don't want to, like, you want to separate the proteins based on their native structure. Okay, now suppose you want to study ligand binding to a protein, or suppose to a calcium binding protein, you want to study, okay, like how much of calcium, what is the concentration or minimum concentration of calcium that can really affect its mobility. If the calcium is binding at a particular concentration, then what will happen is um, its mobility will be affected because calcium has bound to it, and like the native structure or overall structure, if there is any change in the structure of the protein, if there is any change due to the interaction of the calcium ions binding to the particular type of calcium binding protein, then we can determine it on the native page. So in native page, like as the name suggests, we want to uh, like it's exactly same as SDS page, but there are some variations in the type of sample buffer, type of running buffer, and how like what are the running conditions. So what happens there is in SD, just in native page, you won't be adding any kind of denaturing agent because you want to retain the native structure of the protein or the native structure of the sample of your interest. Okay. So in native page, what happens? You won't be adding any detergent like SDS. Okay. You won't be adding any reducing agent. Okay. Because we need to uh, like retain the native structure of the protein or proper folding of the protein. If it's there, or if we want to study the interaction of the legal and the native conditions, then we don't want any influence, like prior influence of any kind of denaturing agent or any kind of uh, detergent, etc., or reducing agent, etc. So you won't be adding SDS detergent, you won't be adding any reducing agent. Okay, whether it's in the sample like loading dye, like yes, sample buffer, or in the running buffer I was talking about, people put tris glycine SDS, right? So the running buffer will also change in case of native page. Then also you native page usually people run it under cold conditions, okay? Or you just can reduce the temperature, or reduce the voltage also, so that the whole setup doesn't get heated, the gel doesn't get heated, because again temperature can also affect the folding of a protein, right? So based on that, so these things have to be kept in mind when you want to run a native page. Okay. So yeah, in the native page, sample is prepared in the loading dye, does not contain detergent or denaturing agent, and as a result, sample runs on the basis of charge by mass. And in the native page, the 3D conformation as well as the activity of the protein remains unaffected. Okay. And there's another thing called urea page. In this method, insoluble protein is dissolved in urea and samples separate based on their charge by subunit mass. A gradient urea page is used to monitor the folding states of a protein. Urea is again a denaturing page, but what happens is when would you use urea page, that you really want to see, like suppose if there are any secondary structures available. Okay, so uh, suppose people run nucleic acids on urea page. Okay, this is also a denaturing page, but you need to like here the like separation is based on charge by subunit mass. Okay. Because suppose things are like some kind of secondary structures is forming, like suppose you are running a nucleic acid, suppose RNA is prone to making different kinds of secondary structures, okay, based on the type of uh, nucleotide available. Okay, now it can form like hairpin loops, etc. Now, but your RNA of interest, your sample of interest is forming a hairpin loop. So definitely due to the structural, like multiple secondary structures, it can move. It will show like a higher molecular weight as compared to its actual molecular weight, right? So what will happen is we adding urea and running it on polyacrylamide gel will dissolve those, help in dissolving those secondary structures, and then we run it on the gel so that we can get an exact idea of what is the exact structure, or if or we can get an idea that okay, if the RNA of our interest or the nucleic acid of our interest is forming any kind of secondary structure or not, right? Because in native page it might run, or on an agarose gel it will run at a higher molecular weight, but on urea page it will run at a lower molecular weight, because the second, in case if it is forming any secondary structures or any kind of hairpin loop, etc., then it will be resolved in in a urea page, and then we can like get an idea, okay, this is prone to forming hairpin loops, suppose. Okay, this is just an example. So urea page can be used for such analysis. Now, so this is an article about urea page, so you guys can go through it. This is an interesting article, this covers everything from sample preparation, why do we use urea page, what all precautions can be taken. Okay, so because we have been talking about the dye and sample preparation for all types of electrophoresis, so in case of urea page, what you do is the dye is also denaturing. It will have um, Formamide, which is again a denaturing agent. Okay, this like especially for nucleic acid samples, formamide can help in denaturing the like resolving the secondary structures. And you add EDTA because EDTA will chelate out any kind of ions and help in protecting your nucleic acid samples from nucleases, because nuclease enzymes require certain divalent cations for its function, and EDTA will chelate those cations. And then you have these tracking dyes, bromophenol blue and xylene cyanol. Okay. Before the samples are loaded, the samples, in case of denaturing urea page, the samples are heated. Mostly it's RNA or nucleic acid samples are heat denatured by heating the samples at high temperatures for like 2 minutes. It will be more than enough. Okay. Now let's go through the slide one by one. So what happens is, especially in case of urea page, the band sharpness depends on several factors. The volume, like some of these things that we will be discussing in this slide will apply in general to all kinds of electrophoresis. Okay. So the band sharpness can depend on several factors, like the volume of the sample loaded. For sharp bands, to get a sharp crisp band, it should be, the volume loaded should be as small as possible, that is ideally between 1 to 5 microliters. However, up to 15 microliters can be loaded, which still ensures acceptable resolution. Less sample volume results in sharper bands, right? Because it will be stacking at a particular level, and then the starting point or the running factor will be same for all the sample wells. Okay. The band quality is also dependent on the gel thickness. For thinner gels, definitely, for like the thinner the gel is, the resolution will be better. Such as 0.75 millimeter show better results than 1.5 millimeter thick gels. The shape of bands can also be influenced during gel loading if the sample is not applied equally into the gel pocket. Okay, so if the density is not good enough, so that's why we add glycerol, as we saw in the previous cases, that adding glycerol will increase the density of the sample, and it will help the sample to go at the bottom of the well. Okay. Uh, Otherwise, what will happen is it will be spread all over the well, spread out all over the well, and as soon as you apply the electric field, what will happen is in the well itself, the sample that is present in the upper portion will first take some time to really come down to the bottom of the well and then start moving through the gel matrix, right? So by that time, another sample that has been loaded in another well, that might be having like really small amount, right? Or it has settled like really well at the bottom of the well, so it will start immediately moving. So this is one of the reasons that we talked about in SDS page also, to have a stacking gel first and then the resolving gel, so that all these samples are almost at the same starting line, so that the race can start. Okay. Now, including 10% glycerol in the loading buffer helps during the gel loading, and the sample sinks into the well more easily. Another problem is if the product runs at the same level as one of the loading dyes. Okay. So what happens is, many times things are fluorescently labeled. Okay. Your sample of interest can be labeled using a fluorophore, and some dyes show a fluorescent signal at a similar wavelength. Okay. Now, if this is the case, removing one dye or all dyes can solve the problem. Then it is advised to run the dye containing sample in an empty well as a size standard. Okay. Also, when you start with a low voltage, okay, at the beginning of the run, it will also help to get sharper bands, as the sample can enter the gel front smoothly and it avoids the gel pockets to collapse due to high voltage. Okay. Another thing is, if you use a short or low percent acrylamide stacking gel, that can have the same band sharpening effect. An often encountered problem is gel smiling, which is due to uneven heat distribution through the gel during electrophoresis. So what will happen is, smile thing is like there's a curvature, right? So what will happen is the same like in the same well, what will happen is the sides as compared to the middle will be heated unevenly. Okay, the heating distribution will be uneven. Based on that, the movement of the protein will be hampered because it will change the porosity of that particular area, right, through which the sample is moving. Also, another thing is if the gel is not surrounded by buffer, if there are no electrolytes in contact, if there is no there is not enough buffer, you haven't really added enough buffer, then you won't be having the movement of expected movement of the electrolytes. Okay, so depending on which gel system is employed, attaching metal plate to the glass plate supports an equal heat distribution and can substantially increase the gel quality. We should also keep a check on the level of the buffer. Okay, many times buffer can get leaked out, and the two sides of the gel should be in contact with buffer, so that, and the buffer should be always in contact with both the electrodes, okay, so that the movement of electrolytes take place, movement of ions take place. Selenizing of glass plates for larger gels is advised since it greatly improves the handling of the gels after the run, as gels tend to stick to the glass plates. So you basically prepare your SDS page on glass plates, right? So it is your gel is actually sandwiched between two glass plates. And then you run it using electrodes. There is the cassette, there is the electrode there, and there is your tank. You put in your apparatus there, and then you start running your sample. But then when you want to stain the things, you want to visualize your protein bands on the gel, you have to take the gel out of the plates, okay, the glass plates. But so care has to be taken when you really are transferring the gel from the glass plates. You have to make sure that the glass plates aren't too dry, because if it becomes too dry, the gel tends to stick to the glass plate. Okay. Also, if we selenize the glass plates, then what will happen is the tendency of the gel to stick, the sticking tendency of the gel onto the glass plate will reduce significantly. Okay, it will come off easily, because many times if it is sticking to the gel, taking out the gel, like as just imagine as thin as 1 millimeter, as thin as 0.75 millimeter, will be like a huge problem, because it helps in breaking of the gel or tearing of the gel. Then you can't really, if things are like torn, then you won't be able to visualize the protein bands. What if like it is torn from exactly at the area where your sample is running? Okay, then it will be difficult to really visualize the things or compare the things on the uh, gel. Okay. So denaturing urea page is useful to analyze or separate single-stranded DNA or RNA fragments, as well as protein samples as well, or some like molecules also, like complex molecules having protein or nucleic acid complexes, that can also be checked on urea page. Okay. Now we talked about protein samples, protein electrophoresis, okay, polyacrylamide element gel electrophoresis. We were talking about the references, right? Protein references, or we also call it as protein ladders. So what exactly are these? Protein ladders are composed of a series of bands which are highly purified polypeptides of different well-defined sizes. Okay. Now definitely get resolved during electrophoresis as separate bands based on their molecular weights. Now there are two types of protein ladders that are available. One is pre-stained protein ladder and another is unstained protein ladder. Now pre-stained protein ladders, what happens is, this is also having multiple polypeptides of defined sizes, but these are already stained. Now what happens is when you are running your sample through, like during electrophoresis, this, like we talked about tracking dye, okay, but this particular thing, this is exactly how you see here on the picture. This is exactly how it is visible on the gel as well when the gel is run. You have to stain the gel to see your proteins or band of sample of interest, right? But this will give an idea that definitely this will, after staining, this will give an idea, okay, if your protein band is here, it will give an idea of the molecular weight of the protein of your interest. But if this will also give an idea, okay, how long should you be running the gel? Suppose um, you know that your protein is around 35, or suppose it is 50 kilodalton. Uh, it has a molecular weight of 50 kilodalton. So you know that your band, you would be expecting to see your band here at this particular point. So you, based on how far this particular lane or this particular marker has run, you can even run it till this part also. Like when I talk about running the gel, it means at the how far this particular dye, suppose this 11 kilodalton is the last one, right? So you can take the risk of moving this sample completely out of the gel only. Okay, because eventually as the samples are moving, as long as you're applying the voltage, as long as electric field is applied, there is a definite dimension of the gel, right? So it samples will start moving out of the gel. So but your protein of interest is 50 kilodalton, so you can take the risk of removing all these bands outside the gel also, and then just maybe stop the thing when this is the last band on the gel. Okay, so just to give an idea that okay, this will also help in tracking how far your gel has run, how far the sample of your interest has run. Okay, so this is the added advantage of having a pre-stained protein ladder. Okay, even before loading the ladders onto the gel for electrophoresis, these ladders are previously stained and ready to go. Because if in case of unstained protein ladder, like your protein of interest, you have to stain it first and then visualize. Okay, then only you will be able to visualize the whole protein ladder as well as your sample of interest, right? So again, all these things will depend on how well the uh, staining solution is. This will also again depend on uh, like many other factors come into play in case of unstained protein ladders, because the staining has to be appropriate as well. And for you to draw a reference, or for you to correctly determine the molecular weight of your protein of interest with the reference, or with reference to the standard marker. So during electrophoresis, the polypeptides of the ladder show up in the gel as distinctly colored bands even before the gel has completely run and then is further stained with the staining dye or staining solution. The ladder bands can be visualized. That is what I was talking about. Okay. And each band represents a molecular weight, which is a combination of the molecular weights of the corresponding polypeptide and the associated dye. This is one disadvantage of using a pre-stained protein ladder, because dye is there, so dye is definitely having an additional molecular weight, right? So it is kind of um, like all these things that has been shown here, this is like nearly this much kilodalton, because it is also having an added molecular weight of the dye that has been added here. Okay. Now see, this is also now another advantage of having a pre-stained ladder is this is the gel that you have run. These are the samples that has run. Okay, pre-stained ladder you have loaded, and during Western blot, what happens is you transfer whatever bands are there on the gel, you transfer it onto the membrane. Now, if it is already pre-stained, you will have an idea that okay, how well the transfer has taken place, or even any transfer has taken place or not. Okay. Suppose you want to optimize the timing, etc., of the protein transfer, you are doing it for the very first time. So you have to check that okay, how well like how long should I put the apparatus for Western blot transfer? Western blot we'll be looking at in the later slides. So how well the transfer has taken place. Brighter ladder bands will suggest a really good transfer, and faint ladder bands, see here in this picture, will suggest poor transfer. Okay. So this is also an advantage of a pre-stained ladder. Whereas an unstained protein ladders are protein ladders where bands or polypeptides are not pre-stained, therefore cannot be visualized while the gel is running. Instead, we can only visualize it after staining with Kumasi or any other staining solution. But because no additional stain is added during electrophoresis, the representative weight of the polypeptide is more accurate because the size of each band of the ladder represents solely the molecular weight of the corresponding polypeptide, because there's no dye and there's no additional molecular weight there. Okay. Now, agarose gel electrophoresis has been discussed like quite a few times. Okay. So agarose gel electrophoresis is a method of gel electrophoresis used in biochemistry, mol bio, genetics, clinical chemistry to separate a mixture of macromolecules like DNA, RNA, or proteins through the matrix of agarose. Okay, so what is agarose? It is a natural linear polymer which is extracted from seaweed and that forms a porous gel matrix by hydrogen bonding when heated in a buffer and allowed to cool. Okay, and this is the most popular medium for separating moderate and large sized nucleic acids and can have a wide range of separation as well. Now it separates DNA fragments by size in a solid support medium like agarose gel. The sample, we looked at it in the previous pictures also, wherein the sample is pipetted onto the sample wells, followed by you apply an electric field or electric current, and negatively charged nucleic acid will migrate towards the positively charged electrode, and the rate of migration is proportional to the size. Smaller fragments will move more quickly and wind up at the bottom of the gel. Okay. Now, how will you, in case of agarose electrophoresis, how will you visualize the gel? Yes, or visualize your DNA of subnucleic acid of interest. DNA is usually visualized by including an intercollecting dye, which is ethidium bromide. This is actually a mutagen, so we have to be careful while handling ethidium bromide in laboratories, like wear proper gloves and all. And then DNA fragments, what happens is take up the dye as they migrate through the gel. And when you illuminate the gel with UV light, this dye uh, gives signal and you can visualize where exactly the nucleic acid band is present on the gel. Okay. Now the larger fragments fluoresce more intensely because it will have more amount of ethidium bromide intercalated. Although each of the fragments of a single class of molecule is present in equimolar proportions, smaller fragments will include less mass of DNA, it will take up less dye, and therefore it will fluoresce less intensely. Okay. And again, in case of agarose gel electrophoresis also, you will have a standard ladder set of DNA fragments of known size that can be run and used to estimate the size of other unknown fragments or your estimate the size of your sample of interest. Okay, this is also like how we determine whether a cloning has really taken place or not. One another like quick way of visualizing is using agarose gel electrophoresis. Okay. Now, in case of agarose gel electrophoresis, the loading dye is again a colored dye that is mixed with DNA samples. So, in the slides, I have been talking about DNA samples. It can be RNA samples as well. Okay. That's mixed with DNA samples before agarose gel electrophoresis to help visualize and track the DNA, or maybe it should be now like any kind of nucleic acid as it moves through the gel. So, what happens is the loading dye makes the sample denser than running buffer, which helps to sink like so that the samples can sink inside the wells during loading. It gives a certain color so that we can help differentiate the sample wells from the gel. Okay, and there's tracking dye also, like there are multiple wells right on a gel, so you can easily track also, okay, which particular well has my sample or which particular well is already occupied. We will be adding it on the next well. Okay, because many times we like there are agarose gels we have to run like suppose 80 samples, 40 samples, 20 samples, and then suddenly if there's no marker or visual marker that okay my sample is there in this particular well, we might end up adding some a very different or some other sample onto the same well where some other sample had previously been added. Okay, so the color helps us to like really see and differentiate the sample wells from the gel. And definitely there's a tracking dye which moves through the gel actually there's a combination of dyes also, which moves through the gel at a similar rate to DNA, so that can be used to estimate how far the DNA has migrated. So see, this is the loading dye. What happens is, there's xylene cyanol, there's bromophenol blue, and orange G. Orange G runs the fastest, like owing to its molecular weight. Uh, so it has been shown that, again, one more thing, when we have been talking about the tracking dyes, okay, the speed of the tracking dyes will vary depending on what is the percentage of the gel, whether it's page, polyacrylamide, gel electrophoresis, or agarose gel. It depends what is the percentage. Okay, so in this particular percentage of gel, 1% agarose and this much concentration of TAE buffer, acha, in case of agarose gel electrophoresis, TAE, or there's tris, this is a kind of tris buffer that is used as a running buffer. Okay, so in this particular concentration of the gel, percentage of gel, orange G has a speed that runs at a speed of around 50 bases long, or nucleotide base pair speed. Then comes bromophenol blue, and xylene cyanol runs the slowest out of these three. Okay, so the dyes composition can include tracking dyes like bromophenol blue, orange G, or xylene cyanol. Many dyes also contain EDTA because mostly you will be running nucleic acids. So EDTA will chelate out the divalent cations that are required by nucleases for their function. So nucleases won't be able to perform its activity and your nucleic acid will be intact. There can be glycerol added to help the DNA or the nucleic acid form a layer at the bottom of the well, and the concentration of the dye can vary and can be adjusted based on the experimental needs. Okay, now I was talking about the running buffer. So there's tris acetate EDTA buffer for agarose gel electrophoresis. There's tris borate EDTA buffer for, so there are different kinds of buffer. The most commonly used ones are TAE and TBE. TAE is the most commonly used one. Okay. Now, when to use what? Okay. The functions of TPE and TAE, this is tris borate EDTA. This is tris acetate EDTA. These buffers include ensuring the current, definitely a buffer should have an electrolyte. Okay. Like should there should be movement of ions, so that ensuring the electric current flows through the gel as well as allowing nucleic acids to move through the agarose matrix. So in addition, the TBE or TAE buffer maintains the pH and the ion concentration during electrophoresis. The pH stays within the appropriate range because the buffer contains weak acid, okay, borate and acetate. Okay, maintaining the pH of the solution is important because the changes in pH might affect the net charge on the nucleic acids, right? And therefore, if the pH is within the desired range, the net charge stays the same and the nucleic acids can move properly. And both tris borate EDTA and tris acetate EDTA buffer contains EDTA, which is a chelating agent which prevents nucleases from degrading the nucleic acids. And nucleases might accidentally contaminate your nucleic acid sample, because especially in case of RNA, what happens, like RNAs are everywhere. Okay, even so, it's always advisable to use gloves while performing your experiments using nucleic acids and all. So what will happen is, it is like on your skin, on your skin, your uh, saliva, etc. will has enough, like, good amount of nucleases. Okay, predominantly RNases are everywhere in the surrounding air and all. So care has to be taken that you really use certain RNase inhibitors in your solutions when you are working with RNA samples. Okay, or EDTA, using EDTA accordingly in your buffers or in your sample solutions can help in preventing nucleus degradation. Okay, so EDTA will prevent these enzymes from degrading your sample even in case of any kind of contamination. Okay, now when to use TBE? TBE buffer is ideal for the following situations. TBE is suitable for obtaining a higher resolution when smaller fragments are you are working with smaller fragments of less than 2 kilobases long, or and using TBE for small fragments provides sharper bands, and TBE has been shown to be to have a higher buffering capacity than TAE, so it works better when runs have to be longer. Okay, when you are running a sample for a longer time, you have to run it for longer time, then TBE has, because it has higher buffering capacity, it will be able to maintain that optimum pH range. Okay, and when should you be using TAE? Now, tris acetate EDTA produces a better separation of larger fragments greater than 2 kilobases long, and TAE works better during cloning because uh, TBE has borate, and borate can act as an inhibitor for some enzymes, because in cloning you need restriction endonucleases, you need ligation, right, of your gene of interest with the vector. Okay, so but using TBE will hamper this thing because it has been found to be an inhibitor of ligase enzyme. Okay, and then TAE works better for performing DNA extraction from agarose gel also. Okay, now again, we talked about different kinds of ladders. Now, for DNA and RNA ladders are also prepared using different kinds of methods, including there will be a plasmid DNA, and that will be chopped, like how they're like using like there were for protein ladders, we saw that there were different sizes of polypeptides, right? Similarly, a plasmid DNA is there, you digest it, a plasmid with a DNA fragment tandem repeat is partially digested with restriction endonucleases. We have to make sure that this particular plasmid DNA will have multiple restriction sites. Okay, so that multiple sizes, like different fragment sizes, are uh, like obtained, so that the plasmid and produce at least 5 different fragments of DNA fragments of different sizes. Okay, then what you do is you will have different sized fragments. Now you have, because that much of fragment won't really be helping you in uh, quantifying the uh, not only quantifying, or because we have to use that particular marker multiple times, okay, for multiple runs, for different kinds of scenarios, for different kinds of experiments. So you should have enough amount of the marker present. Okay, so definitely you have to, when you have got different sizes of your DNA, okay, then you will have to amplify the number of uh, fragments. Okay, uh, like the fragment size of each sized fragment, you have to amplify it so that you have enough amount. So you carry out polymerase chain reaction, or PCR, and DNA fragments can be prepared using PCR amplification of desired bands, followed by mixing the purified PCR products at specific concentrations, and then you combine PCR and restriction digestion. A combination of PCR and restriction digestion can be used to prepare DNA ladders, as we saw above. Okay. So when you are actually choosing a certain type of DNA or RNA ladder, or you are about to prepare one, suppose, okay, what should be considered? What are the different kinds of things to be kept in mind? What are the number of bands? We saw here that it should at least have 5 different bands. Okay, like five bands of different sizes. Okay, and ladders with more bands can make it easier to estimate the size of fragments in a test sample, but the gel needs to run longer to differentiate the bands clearly. Now, one more thing, like we looked at uh, suppose this ladder, protein ladder, or in general any kind of DNA ladder or RNA ladder we are talking about, this separation should be optimum. Okay, when you start, if you when you start running the gel, what happens is these are like extremely compressed because this hasn't separated, because it has been stacked at a particular point. Right now, over the time it will slowly resolve and eventually separate properly. So this gap should be proper, so that you are able to determine accurately, or like almost accurately, the size of your sample of interest. Okay, whether it's a protein or a nucleic acid. Okay, So this consideration should be there that the gel needs to run longer to differentiate the bands clearly. Ladders with fewer bands may be better if time is limited, but only a rough estimate of band sizes is needed. And buffers containing tris, acetate, and EDTA are commonly used for ladders with larger fragments, while those containing tris, borate, and EDTA are typical for ladders with smaller fragments that we looked in the previous slide. Okay. Now see, this is how like an ideal DNA ladder should have several characteristics which the electrophoresis experiments need. Every fragment of the ladder must be separable from each other, right? This is how it should be. And every fragment should have enough concentration so that we are able to visualize the ladder at least on the gel. Otherwise, what would we have to really compare it with? Compare our sample of interest with. Okay. There should be a reference and the reference should be visible clearly. The composition of DNA gel loading dye must not affect the specificity of the DNA ladder, and the ladder must be definitely it should be stable enough and should have enough quantity to use for a longer period of time, and the ladder must be highly purified. So this can also be people can incorporate chromatography based purification techniques to purify the different fragments. Okay, so basically the underlying statement is it should be highly purified. Okay. Now these are some of the things we can do or the troubleshooting we can do during molecular weight determination. We talked about the smiling bands, which takes place due to uneven heating of the gel, thereby leading to differential migration of proteins or nucleic acids, okay, with outer lanes moving slower than the middle lanes. This is why this thing is there, that curvature is there, okay. Then there can be diffusion. It not only happens in case of protein bands, it also happens in case of nucleic acids. Diffused pattern of the protein band appears on the page. Diffused protein bands can be corrected by increasing the running current, okay, or higher concentration of acrylamide. So here, all these things have been talked about with the consideration that the sample of your interest is having proteins. It is a protein. And there can be vertical streaking. Vertical streaking of the protein bands can appear due to overloading of the protein sample. Either you can reduce the amount of protein sample or you can run the gel at lower current so that the whole sample can stack at a particular point and there is no abrupt shift into the resolving gel. Okay. And protein is running faster than expected. Sometimes the migration of protein is not proportional to the molecular weight and it is either more or less on gel than the expected place. It is due to the unusual high proportion of basic or acidic amino acids. Okay, sometimes or many times it might happen that suppose your protein of the band of your protein of interest is appearing slightly above. Okay, suppose your protein is 10 kDa, but it is on the gel you are seeing that it is running nearer to 12 kDa like that. So based on what is the amino acidic, like what is the composition, amino acid composition of your protein, based on that, because amino acid will have a net charge on it, right? That we looked at like glycine. But then there are glycine has only H, right? But in place of H, there are other functional groups in case of other amino acids. So those functional groups will eventually impart certain charge uh, to the overall protein, okay, which can affect the movement in an electrophoretic system. There can be double bands. Double bands can indicate that there has been degradation of the protein sample. So addition of more amount of reducing agent, we can add more reducing agent or prepare a fresh sample to reduce these artifacts. Protein bands can be distorted. Again, if the protein bands are distorted, uh, stacking gel, it can be corrected by increasing the amount of APS or timid or de-aerating the stacking gel. Okay, one more thing. Like we saw, its resolving gel is there, there's stacking gel. So, the more, like we have added APS and timid. APS gives free radicals and timid catalyzes the polymerization reaction. Okay. Now, if the gel is exposed to the oxygen, what happens is there might be hindrance or interference in the polymerization process, overall polymerization process. So what is the best method? What people usually do is, like once you add the stacking gel on top of resolving gel, you add a layer of isopropanol, okay, or like some alcohol which cuts off the supply of oxygen to the gel, okay, and prevents the exposure of that stacking gel to the oxygen or surrounding oxygen in the air, and helps in smoother polymerization to take place. Okay. There can be lateral spreading also. If the protein bands appear laterally spreading, it can be avoided by reducing the time between loading of the sample and running. Lateral spreading means it has started diffusing. Now let's talk about Western blot. It's the same thing. You load your sample, you let it run, electrophoresis there. And now what you do is you transfer, this is the gel here. Okay, this is the membrane wherein all these bands have to be transferred so that you can identify your protein of interest using uh, uh, any kind of antibody, or like which can have some kind of, like using chemiluminescent signals, fluorescent signals, or there can be antigen antibody interactions as well. Okay, based on these interactions, you can detect whether your protein of interest is present or not, suppose in a complex or in a particular lysate. Okay, so transfer methods can be wet or semi-dry. Wet is wet, wet transfer methods basically is here, this particular part is completely present inside a buffer, whereas in semi-dry technique, you just have to uh, just let these things like soak in the appropriate buffer, and very small quantity of buffer is required in case of semi-dry transfer, and semi-dry transfer takes like 25 minutes to 30 minutes, like really less time as compared to a wet transfer. It takes longer. Okay, a wet electroblotting takes longer, and semi-dry blotting takes like max to max an hour. Okay, so, and semi-dry blotting is better suited for lower molecular weight proteins, whereas wet electroblotting can be used for a range of molecular weights, for transferring low to high molecular weight proteins more than 100 kDa. Okay, but it is time consuming, as I was talking about, and typically requiring nearly an hour to overnight. It can be 2 hours, it can be overnight transfer, okay, based on what is the sample of your interest. So this is the overall um, like apparatus, or this is the overall setting. This is the direction of the current and protein migration. So what you do is, this is the transfer cassette. This is the negative one. This is the positive one. In between, you apply some foam pad and filter paper so that there is even, what will happen is, if you directly put, the gel is very fragile, right? So it needs a support, proper support to really, um, and also sorry, the filter paper here is also fragile as compared to, you can't really directly add a filter paper because it is fragile and it won't be able to maintain that particular setting. Okay, so a foam pad which is a bit sturdy, as well as it will allow the buffer or whatever solution the surrounding medium uh, needs to be provided. This foam pad will eventually be able to let the solution pass through. Okay, so these boundaries of foam pad and filter paper should be there, and then this should be taken care of like really properly. The gel should be towards the negative side, and membrane should be towards the positive side. Why? Because the protein bands and whatever protein bands are there on the gel, that will be transferred onto the membrane. Movement of electrons, due to the movement of electrons from this area to this area, like this cathode, this electrode to this electrode, will eventually lead to the transfer of the protein bands from the gel to the membrane. So this movement of electrons will do that. So what will happen is, this If you put the membrane towards the negative side and gel towards the positive side, no transfer will take place. Okay, basically everything will go out. And also you have to really optimize the transfer time, because if the transfer time is more, eventually what might happen is protein can like the protein can come out into the solution as well. Okay, in case of wet transfer. Okay, although it's like really rare, but in extreme cases or some extreme scenario, this might happen. Okay, now membrane selection. Usually two types of membranes are used. One is nitrocellulose and one is PVDF or polyvinyl difluoride. So nitrocellulose can immobilize proteins and glycoproteins with high efficiency via hydrophobic interactions, but it cannot be stripped and reprobed many times. So reprobed means we are talking about antibody binding. Okay, we let the membrane incubate use we incubate the membrane using a solution of antibodies, right? So what happens is the nitrocellulose membrane can't be reprobed, but the polyvinyl fluoride, once you have used a solution of antibody of a particular protein, you can again use it using another like again probe it using a different kind of antibody. It will give better results in case of PVDF, but not in case of nitrocellulose. PVDF, but what happens? PVDF has a high protein binding capacity. PVDF is highly hydrophobic and must be pre-wet with methanol prior to use. Okay, but it is less brittle than nitrocellulose. PVDF can be reprobed many times. Both nitrocellulose and PVDF are available in a range of pore sizes. While 0.45 micrometers pore size membranes are generally applicable, 0.1 to zero point 0.2 micrometer pore size membranes are more suitable for blotting low molecular weight proteins. Because again, I was talking about proteins coming out of the membrane as well, depending on how much size is the how much pore size does the membrane have. Okay, so then what happens is after your protein transfer has taken place, you uh, block the solution. What happens is this is usually either skimmed milk solution or a kind of TB like tris buffered solution. Like you use either skimmed milk or BSA, where you actually try to cover the membrane to prevent any kind of non-specific binding, so that the you will be adding primary antibodies, right? You'll be adding primary antibodies, and the primary antibodies should not bind to any other area of the membrane. It should bind only to the proteins that are exactly at the regions where the protein bands are present. Okay, so blocking, you use either skimmed milk or BSA, and then you probe. Now in Western blotting, there can be two ways of probing. One is two-step probing, one is one-step probing. In two-step probing, what happens? Membrane is first probed with the primary antibody to recognize the protein of interest. In like the antigen here is the protein of interest, and primary antibody will bind to that. And membrane is probed with primary antibody having an appropriate dilution for a particular time, and then you add secondary antibody. Okay, it is reprobed with another antibody directed against the primary antibody. Secondary antibody is conjugated with a fluorophore or a particular enzyme. Okay, when we add if it is conjugated with an enzyme, then you have to add the appropriate substrate and then let the chemical reaction take place. Suppose a luminescent substrate is there, so it will emit the particular, we can get the signal after the reaction has taken place. So based on that, so there are, you are using two-step probing is you are using two different antibodies, which increases the sensitivity as well as giving flexibility to plan multiple probing, right? And one-step probing is just you are adding primary antibody against which will identify only the protein of interest, and the primary antibody is tagged with a fluorophore or an enzyme. Okay, one, and this is not really common because what happens is this might like tagging the antibody with the fluorophore, the signal, the eventually what how much signal you are getting that will decrease in case of one-step probing as compared to two-step probing. The amplification of the signal will be much higher in case of two types of antibodies, primary antibody and secondary antibody. Okay. So yes, like this is a colorimetric different. What is this reagent here? What is the, whether it's enzyme based, whether it's a fluorophore based, whether it's a luminescent signal being given out. Based on that, there are multiple ways to detect what type of molecule has been tagged onto the secondary antibody. Okay, for detection. So now let's come to the question answer. Like it's like very direct questions here. So first question is electrophoresis of a molecule is carried out in a solution, not a solid matrix. In what kind of electrophoresis? So what will be the answer? Yes, it is moving boundary electrophoresis. You don't have any like supporting medium or supporting matrix. Okay. So in case of page, what is the initiator for acrylamide polymerization? Yes, it is it supplies the persulfate, okay, or like radicals, free radicals. Now the progress of a protein sample in a page gel can be monitored by, yes, bromophenol blue. And CBB is chromacy brilliant blue, which is for detecting your protein bands. SDS is the detergent. And does anyone remember what phenol red does? Yes, yes, very good. Like it's a pH, basically an indicator of the pH of the medium. Okay. Now, which of the following components imparts a negative charge to a protein? Yes, SDS. Now in pH, a layer of organic solvent is introduced on top of the resolving gel or stacking gel. It can be stacking gel also. So what is happening? I just talked in few slides back. Like we add isopropanol, a layer to cut off the oxygen's interaction. Okay. So that polymerization can happen properly. So this layer of organic solvent, which can be usually it's isopropan or like any kind of alcohol, we use it to prevent the entry of oxygen. Okay. The pore size in a polyacrylamide gel can be controlled by manipulating the ratio of, yes, acrylamide and bisacrylamide. So, what is the branching or cross-linking agent out of these two? Does anyone remember? Like one is forming the, yes, 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 very good. Okay. Now, which of the following component in the loading die increases the density of the protein sample? It can be protein or any other nucleic acid sample and facilitates its loading into the well. So, yes. pH of the stacking gel is 6.8. So, the protein front is sandwiched between which two layers or which two fronts? Yes. Now, which of the following can be used to monitor the folding states of a protein? Yes, yes. So this is the gradient urea page. Now what happens? We saw that at different kinds of urea concentration, the protein folds eventually. So what people usually do is they can make urea gradient so that there's a gradient of urea, like suppose from no urea to high concentration of around 8 molar urea at these wells. Now, usually you have seen in the demo videos also, the gels are made like this. But what happens is you just invert the thing, not really invert, just maybe rotate the thing by 90 degrees, and then start adding your like each solution having like 0 molar urea, 1 molar urea, 2 molar urea, like that, one by one, and then let the things diffuse amongst each other. But then this particular, then you just keep it straight, and then the this one will have the highest concentration of urea, and this will have lowest one. So the unfolded protein will be like maximum unfolding, or probably like complete unfolding will be visible towards this extreme right portion. And there, and natively folded proteins, because being natively folded, it will move the fastest. So the bands will be seen at a lower area as compared to, like, lower region as compared to the bands seen here. Okay, like this is unfolded protein, this is for the native protein, because this region has higher concentration of urea. Okay, now we talked about some troubleshooting techniques in page. So vertical streaking of protein bands appear usually due to, it is due to overloading of protein. When you load a lot of protein, what might happen that it will take time for the sample to really reach the uh, bottom of the well. Okay, so you can see like some of the protein might still be there, like, and most of the protein have like has entered the resolving gel, like separate, and separation has started. But because you have overloaded your sample, many of the, like the layer on the in the sample well that is present near the top, that is still taking time to really, that has taken time to really get into the resolving gel. Okay, and so you see that vertical streak of protein bands. Okay, now this is kind of the same problem we saw the earlier week also. The molecular weight of a protein in its native state is 240 kilodalton, and on SDS page, which is denaturing, it is 60 kilodalton. So protein is not a heteromer, like not like a hemoglobin. It is a homomer, like all the subunits are the same. So what is the oligomeric status of the protein? Option option D. Yes. Good. It is tetramer. Okay. Oh, sorry. So the electrophoretic mobility of a protein in urea page decreases due to, like, the unfolding. Yes, it increases because the protein is unfolding. Okay, increase in hydrodynamic volume. And this is the last question. So in the West, we talked about primary antibody and secondary antibody. Okay, so suppose this is the protein that is being probed, and the primary antibody used is a mouse generated primary antibody. So the secondary antibody is conjugated with this particular enzyme. This is a peroxidase. So what should be the type of secondary antibody? Does anyone does anyone have any idea? Yes, yes, because what happens here is primary and any antibody that is being generated, you have to really culture these antibodies in certain organisms. Okay, now if your any antibody will, this particular primary antibody is mouse generated. Okay, so whatever thing, like, has been in the mouse, whatever is antigenic for the mouse, that will be uh, acted upon by this antibody. Okay, but you can't really generate a secondary antibody in the mouse itself. It should always be a different organism, because then the primary antibody won't be really recognized as an antigen here. Okay, the secondary antibody should be generated in a different organism. So it should be anti-mouse, because otherwise, if the suppose secondary antibody has, if you try to generate it inside mouse only, then the primary antibody is like compatible with the mouse system only, right? It is helping the mouse in surviving, kind of. Okay. So the secondary antibody won't be able to recognize the primary antibody, because primary antibody is being used by the mouse, right? Or suppose if secondary antibody against primary antibody is developed, then it will eventually like uh, take up the or degrade the whole primary antibody system, right? Like whatever has been developed against this particular antigen. Then the whole point, like whole thing, whole system doesn't make any sense, right? So the secondary antibody should always be against primary antibody. So what happens is this primary antibody that has been developed in a particular organism. In this question, it is mouse. It can be rabbit. It can be horse. It can be goat. Okay. Now this primary antibody is injected into another organism, and then if it is injected into another organism, then that will act as an antigen in that particular system, and the other organism will develop antibodies against this particular antigen. So for the other organism, the antigen here is the primary antibody developed in mouse. Okay. So here it should always be anti-mouse. Okay. The thing that you should be looking for is anti-mouse. The organism can be goat, rabbit, okay, anything. It can be horse, goat, rabbit, anything, but it should be anti-mouse. Okay, and this particular HRP, it can be any other fluorophore, it can be HRP, whatever like molecule is conjugated based on what type of detection technique you are using. Based on that, this conjugation should be there. But what in case of questions like these, you should be careful about this particular part, okay, which is the organism where primary antibody and secondary antibody has been like cultivated or cultured. Okay, so answer is horse anti-mouse HRP, because primary antibody was generated in mouse. Okay, okay then. This is it for today's class. We will again meet the next week. Thank you for joining. Yes, yes. Yes. It is MCQ based only, like these only, like MCQ based. No, it will be like ranging towards this concept based only. It will be like familiar questions only. Yes. And also, if you have any questions, you can always bring it in the next class also. We can discuss it. Or suppose if I am unable to answer at that particular point, we can discuss and come up to a solution also if you have any doubts. Okay, we can always discuss that. Okay. So when is the exam? Like I think it's October, right? 26 October. Okay, okay. Okay then. We'll meet in the next tutorial session. Okay. Yeah. Thank you for joining.